Good morning, ladies and gentlemen. Welcome to the visiting lecture of Environmental Science with Dr. Marta Ujevodka from Institute of Geological Science, Police Academy of Science, Poland. Before we start the sessions, I would like to first welcoming Dr. Marta to the event. Good morning, Dr. Marta.
Good morning
to
everyone.
Yeah, we can hear your sound clearly, and also
Good.
your video is also okay. So
Good.
we assume that the internet connection is stable at this moment.
Yeah.
And we also would like to see if Professor Widowati is present. Professor Widowati is already present before.
Yes, 2 seconds.
Internet connection is a bit problem this morning, so I will check again. Yeah, oh, not yet.
Hello, I kick out and then I join with
Okay, Prof. Nani.
Would it connect
yes.
now?
Thank you very much, Prof. Nani.
Well,
Okay, Prof. Widowati is also present now. Okay, then we can start the program. Okay, Bismillahirrahmanirrahim. Good morning, ladies and gentlemen. Welcome to the visiting lecture of Environmental Science, presented by the Faculty of Science and Mathematics, Diponegoro University, Semarang. On today's lecture, we have Dr. Marta Ujevodka Prisbit from Institute of Geological Science, Police Academy of Science, Polandia, as a speaker. Dr. Marta will give a lecture about lake sediments as an archive of environmental changes case study. So, my name is Panji Tridiaksa. I will be the host of this event. And before we start the session, I would like to kindly ask and invite our Dean, Professor Dr. Widowati, to open the lecture. Please, Professor Widowati, the time is yours.
Thank you very much, Mr. Panji. Assalamualaikum warahmatullahi wabarakatuh. I hope all participants can hear me.
Yes.
Mr. Panji, you
Yes,
can
yes.
hear me?
Professor.
Yes, okay. Yes. Okay. Good morning, everybody. Excellency Marta Ujevodka, PhD from Institute of Geological Sciences, Police Academy of Sciences, Polandia. Good morning, Dr. Marta.
Good morning to everyone. Thank you.
Thank you very much for your joining in this event. Excellency Farihin, PhD, as a Vice Dean for Academic and Student Affairs of the Faculty of Science and Mathematics, Diponegoro University. Professor Dr. Kuswara Adi, as a Vice Dean for Human Resources, and also Professor Dr. Tri Retnaningsih Suprabhawati as a moderator. Honorable Panji Triadaksa, PhD, as a Head of International Office, Faculty of Science and Mathematics. Dear lecturer and all participants, it is my great pleasure to cordially welcome you to the Faculty of Science and Mathematics program visiting lecturer events. First of all, let us thank Allah Subhanahu wa Ta'ala, the most gracious and merciful God, because with His permission today, we can gather here to join together for online visiting lecturer events. My sincere gratitude, especially to Marta PhD, who will be today's speaker, and to all participants for their great contribution in promoting sciences and technology for Diponegoro University Academia. In addition, for today's lecture, the topics of the material is Lake Sediment as an archive of environmental changes, a case study. It is an hour for us to get a direct lecture from Marta PhD. Ladies and gentlemen, Diponegoro University is one of the leading university in Indonesia, as well as the Faculty of Science and Mathematics, which is one of the best faculties at Diponegoro University. As we know that Diponegoro University's significant achievement and development can be seen through the exponential growth of science and technology through innovation research, increasing number of international publications in highly reputable journal, and also internationalization activities. Therefore, we continue to improve our ability to become a research faculty with international reputation. It is in line with our vision. To achieve our vision, one of them is visiting lecturer activities. With this activity, we hope that the speaker can share experiences and share knowledge with all participants. In the near future, I wish that the Faculty of Science and Mathematics can collaborate with Institute of Geological Sciences, Police Academy of Sciences, Polandia, through activities such as joint research program for academic staff member. Of course, this topic might include research related to biology, physics, chemistry, computing science, environmental science, information science, mathematics, and life science. And also, I hope we can collaborate, join publication, join seminar, on join workshop, and also academic staff and student exchange. Last but not least, by the grace of God Almighty and consent of all participants, on behalf of Faculty of Science and Mathematics, I am, as a Dean, hereby announce that online visiting lecturer events with the topics Lake Sediment as an archive of environmental changes, a case study is open. I encourage all participants to participate actively in the interesting discussion. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Dr. Widowati, as Dean of the Faculty of Science and Mathematics, and also for opening the session. And for continuing the sessions for this morning, we would like to invite Dr. Marta Wojewodka as a speaker, and also Professor Dr. Tri Retnaningsih Sukabawati, Master of Application Science, as a moderator. I would like to introduce Professor Dr. Tri Retnaningsih. She is a professor as a professor from the Biology Department, Faculty of Science and Mathematics, and she also a lecturer at the School of Graduate School of Environmental in our university also. And to proceed the sessions, please, Professor Tri Retnaningsih, to continue the lecture. Good morning, Professor Tri Retnaningsih. Or it seems we have internet connection problem.
Yes, sorry, give us a second.
Okay. Yeah. Because Professor Tri Retnaningsih and Dr. Marta is at the same room, so they chat with each
Yes.
other. Okay.
Sorry, Pak Panji, that we always kick out, and then now hopefully it is working well because
Okay.
somebody, guys already help us to join in Lantai 2 connections.
Okay, thank you.
Okay,
please
Marta still preparing everything because she has to start it for the beginning. But while waiting for Marta, I will. Can I just introduce Marta first? Pak Panji, because I kick out whenever you speak. Okay, okay.
Yes, please.
Okay, yes. Thank you. Assalamualaikum warahmatullahi wabarakatuh. The lecture series for today is part of the activities that will be done during Marta's stays in Undip as a visiting scientist, supporting by world-class university program of Undip. And then we have a lot of work that already done 2 days ago. We have workshop on identification on Cladocera, and thank you, Dean of Faculty of Science and Mathematics, as well as Vice Dean 1 and Vice Dean 2, as well as all staff at FSM that supporting our workshop 2 days ago, since 2 days ago, and the participant is so high, and we appreciate to that. And for today, it's still relating to the expertise of Marta, that Marta will deliver about lake sediment as an archive of environmental change with a case study. Let me introduce Marta first. Marta Wohewotka Prispil, I hope I spelling a little bit right now, is expert on paleolimnology, especially on Cladocera. Although we are in the same paleolimnology, but we use the different organisms and strengthen each other. And her PhD is on the earth science, and then she will study PhD as well in paleolimnology, and then MSc in hydrogeology, and for bachelor on oceanography. And she has many works experience as an assistant professor, and then research assistant, technician, even internship, and the freelancer and voluntary work at Ministry of Environment in Poland. And then she has a lot of awarded, success honor of a PhD thesis awarded, and then award for the best PhD student at IGS PAS Warsaw, and then also twice in 2017 and 2018. And then last year, she was have a fellowship and award at Department of Ecology and Biology, University of Matej, Slovakia. And she has many projects, mostly, of course, on paleolimnology. So for today, she would like to share her experience related to the to use the lake sediments to reconstruct past conditions, and of course, whenever we already able to have known past conditions, able to use as a base for lake management. So, Marta, are you ready? Okay, okay, time is yours. Okay.
Okay, thank you very much for this introduction, and good morning again to everyone. I'm happy that that you join us, that you decided to join us. And let me share with my screen. Do you hear me well, by the way?
Yes.
Okay. So, as Professor already mentioned, today I'm gonna to talk about the lake sediments as an archives. Do you see my presentation? Yes, good. So today we are going to talk about the lake sediments and their roles as the indicators of the past environmental changes. And the main part of my presentation will concentrate on the case study I would like to share with you. But let's start from the beginning, let's say. So all the lakes have changed over the time. So this, as each ecosystem, lakes are like the infinite network of the interactions between among the biotic and abiotic components of this ecosystem that are balanced by like the internal processes and also by external factors. So when this interaction are balanced, the ecosystem is quite stable. When the when the interaction become unbalanced, the ecosystem start to change. And these changes, of course, can be rapid, small or substantial, can be rapid and very profound. Of course, when we are talking about the rapid changes in the present times, we can monitor such changes through the regular observation. However, many changes of the ecosystem have been proceed over a long term of the long period, far back before the regular observation have been possible. So also some short, very profound and extensive changes have happened also far back in the past. So how we can know what kind of processes affected our ecosystem, causing that this ecosystem looks like this nowadays. And with the help, it's coming the paleolimnology, it's the scientific disciplines that cover an area where ecology of lakes, of course, limnology and paleontology, so the study of the microfossils meets. And usually the lakes, the paleolimnology study the origin and geomorphological history of the lake basins, but also the response of their biocenos, so the plants and animals that live there, in their response to the external and internal processes I already mentioned. So how it's possible? Actually, everything what's happened in the catchment of the lake and everything what's happened within the lake, it's recorded in their sediments. So by studying the lithological characteristic, by studying the changes in the geochemical composition, as well as like studying the plants and animals remains, we can reconstruct the changes of given ecosystem through the time. So how we doing that? So with the time, at the bottom of the lake, the sediments are deposited, and each sediment, each layer that is deposited is corresponding with the very specific time in the past. But before details about the paleolimnology, I would like to also mention how we can get such core. here you have an example of the core collected during the paleolimnological study. So usually to get like the short cores, so similar to this one, we are using so-called gravity corer, and you have several types of the gravity corer. Let's see how it works on the movie of the on the movie provided by a company Ecotechnica on the YouTube. So let's look at. So this corer is this Uvitec corer, one of the type of the gravity corer, with which we usually can take rather short cores. And then usually in the field, we are cutting the obtained core by this kind of the equipment into the subsections, the small layers of the cores. And each layer, of course, is linked with the different time in the past. So this was example of how to collect the short core. And of course, if you would like to, I didn't mention, sometimes also we, especially in the high mountain lakes, we did it also in the Europe, in temperate zone. We can collect the sediments from the ice. So it's much easier because you don't need boat. So here you have the example how it looks like. And this is picture done by my colleagues from the institute. Of course, if you would like to obtain the long core, you need more advanced equipment, and it's also, of course, much more expensive in time and funds, of course. Okay, what's then? Of course, the core, the uppermost layer of the core, if it's not mixed, should correspond with the youngest period, and the bottom of the core should correspond with the oldest period. And after obtaining the core, we need to divided our core into some intervals. Of course, smaller, or the tinier layers of the sediments will related with the shorter time intervals, so we could obtain the like high resolution of our research. But of course, we need to remember that it will also be such studies will be more time consuming and also will be much more expensive. And after that, when we subsectioned our core, we need to obtain the so-called age depth model. So for that, so we need to know exactly at which time the given layer in our core was deposited. So for this purposes, usually in paleolimnology, we are using radioactive isotopes, especially of carbon. So, okay. Especially of carbon, so C14, and this method is used usually for the older sediments, like the maximum 50,000 years. And for the youngest sediments, usually in paleolimnology is using isotope of lithium, of lead, so PB 2010. Sometimes paleolimnologists are lucky and can find the annually laminated sediments. And in such case, the one dark and one light layer corresponding with one year of deposition So by counting these layers, they can obtain very high resolution reconstruction of past conditions. And also sometimes, especially in case of the older sediments, there is also using tephrostratigraphy. So each volcano eruption have different chemical fingerprints. So the position of the ash in the lake sediments can allow us to correspond the specific layer with the specific event in the volcanic activity. So to sum up how the paleolimnology methods, what is the background of the paleolimnology method? It's firstly, we need to collect the sediment core from the bottom of the lake or the other water body. After that, we need to subsectionate our core. The crucial step is, of course, as I mentioned, dating of the core, so relation of the age to the depth, and some geochemical analysis. Another step is analyzing of the so-called proxy indicators. So these are usually remains of the plants or remains of the animals that the knowledge about their current requirements allow us to reconstruct the changes of the environment through the time. So information about the abundance, species composition, and so on, we can draw on the stratigraphic diagram and then reconstruct the environmental composition, environmental changes. Sorry. So now I would like to take you and show you one of the example of such study. So I would like to take you to the Central America, to El Salvador, and so we are in the northern hemisphere. And this study were done with collaboration of the fantastic people that we already published the paper about this study. So as you can see, this is a big team. So everyone was responsible for different proxy analysis. And we studied the case study I would like to show you today. It's the Lake Apostatec. It's Mar Lake, located in the central El Salvador, quite close to the San Salvador. This is like the like 40 kilometers, and it's located at the altitude above 500 meters above sea level. It's quite small lake. But as it's typical like for the Mar Lake, that they are small and quite deep. So the maximum depth of the Apostepec Lake is around 50 meters. This is the lake with the low conductivity, with it's alkaline waters, and dominant ions are bicarbonates and calcium. And in the waters, we can also find quite high content of silica, but this is normal because for such lakes, because the lake is surrounded by basaltic ash and andesite blocks. So this kind of rocks have quite high content of the silica. As I already mentioned, this is the Mar Lake. So it was its origin is related with the phreatic eruption, and as you can see on this picture, this lake is surrounded mainly by the croplands and some agricultures. And it's located within Apostepec volcanic field, where a fault zone extends toward the east. And this is in the landscape. It's displayed by semicircular scarp. Near to the lake Apostepec, it's Lake Sieg. This is very shallow lake. The important for our story is to mention that in the past reports of the Ministry of the Environment, the Apostatec, they reported that the size of the Apostatec, the surface area of the Apostatec lake is changing during the rain season and double its size. Nowadays, the newest report, in the newest report, such information are not present, but they mentioned that the Lake Sieng during the rainy season, flooding the all agriculture and croplands around the lake. So we cannot exclude that actually in the past those two lakes were merged. And this is further supported by hypothesis of the researcher from the El Salvador that the Lake Sieng is like the remnants of the bigger lake. The climate of the region is quite similar to the climate in the Java actually. So we have two seasons, like the rain seasons and dry seasons, but because this is on the northern hemisphere, so the dry season started at the October and finished in the April more or less. So this region is characterized by a bimodal cycle of precipitation, and this is caused mainly by so-called intertropical convergence zones. So movement of the intertropical convergence zone during the dry season into more southern position is resulted in the lower precipitation. And of course, the most variable climatic elements in this region is rainfall, which is, as I already mentioned, by intertropical convergence zone. Okay, you can ask me why actually this region. This region is important because 7% of the world's biodiversity is hosted in by Central America. So it's good to know, it's important to know the species composition, the responses to the environmental changes, the sensitivity to the climate and environmental changes. This is also important region because there is still very limited information about the past, there are still limited records of the past climate changes and past environmental changes. And this is important in the El Salvador, especially because this country is overpopulated. In the 19th and 20th century, almost 90% of the country was deforested due to the development of the commercial agriculture. And of course, there is in the lake is big pressure that comes from the fish farms. So by using the bioindicators, we were trying to track the effects of the recent climate oscillation, so-called Little Ice Age. This is the last global cooling that were recorded over the world, but was not comprehensive. And in some places, we could obtain the different signals of the climate and environmental changes. And also we were trying to see how the recent human impact and also geological events like the earthquakes affected our lake. So for that purposes, we, as I mentioned, obtained a short course, 34 centimeters long, that we split it into 2 centimeters sediment layers. In each sediments, of course, we dated our core. For each layers, we analyzed several biological proxy like the ostracods. So these are small crustacea, like the chironomids. So this is larvae, like the cladocera. So these are small microcrustacea, diatoms. So these are algae, example of the algae, and amebs. And also this biological analysis were further supported by the geochemical analysis. And as you can see, the sediment core is quite homogeneous. So it has like the fine-grained texture. But actually for the Mar Lake, it's quite typical because, at the zone depth at the bottom. So we usually have like the clay sediments. Yeah. And we did also age depth model based on the PB dating, so lithium dating, carbon dating, and the dating displayed that our core cover like the last 500 years. As I mentioned, in the results of the, we did the several biological analyses, and for each bioproxy, we obtained stratigraphical diagrams, but it's quite difficult to analyze each of separately. Also for geochemistry, we obtained the stratigraphical diagram of changes. To make our work easier, we just make one summarizing diagram on which we put only the most important indicators from each group of the proxy we analyzed. And the bottom sediments, so the first layer, as I mentioned, it's the core span the last around 500 years. So the bottom sediments likely correspond with the beginning of the Little Ice Age. It's possible that also with the Spurer minimum. Spurer minimum is the minima in the solar activity, which caused the decrease of the temperature. And at that time, we observed quite high share of the planktonic cladocera along with the planktonic diatoms. And this indicates actually that environmental conditions at that time was characterized by high water level. And also, when we look at the diagram of the such indicators like total organic carbon in the sediments or content of the phosphates, we can deduce that at that time the trophy state was relatively high, let's say. So, yeah, here you have the higher. You have these geochemical indicators. Also, in case of the total nitrogen, we saw like the higher content than in the following, in comparison to the following zone. We also observed the low values of the indicators of ratio iron and manganium, and this ratio is usually in paleolimnology using to talk about the changes in the redox conditions. So it means that the water column could be quite well oxygenized. However, we are thinking that it was this was caused by rather higher volumetric ratio of epilimnion versus hypolimnion. Epilimnion, so this is the layer in the lake that is rich in the oxygen, while the hypolimnion, so the bottom layer of the water column, it's usually poor in the oxygen. After that, we can observe the gradual increase share of the taxa in the taxa that are related with the benthic habitat and the littoral habitat. So littoral habitat is the shore zone, so the shallow water conditions. So it could suggest actually that the lower water, decreasing of the water level. At that time also we observed a drop in the total cladocera and chironomid abundance. And yeah, and also at that time we can observe higher share of the higher values of the ratio of chironomine and tanipoidine. These are groups of the chironomids, and increasing higher values for this ratio may indicate the opposite scenario, like the increase of the water level. So from the biological proxies, we obtained two different scenarios. So from the diatoms and Cladocerra, we gave information that there were rather drop of the water level, whereas in case of the chironomids, we got information that there were rather increase of the water level. So finally, we took into account the we took into account the fact that this is Mar Lake, so this is the lake with very steepy walls, and we took into account also the fact that in the past reports of the Ministry of the Environment in El Salvador, they reported significant changes in the surface area of the lake. And also we observed higher content of the alkaline metals. The alkaline metals under more humid conditions may be leached from the rocks into the lake. So it supports rather the scenario of the higher water level as a result of higher humidity. So it seems that the scenario number 2 is more likely in this case. Yeah, so this is what I already mentioned, that we observed at that time increase of content of the alkaline metals. Yes. And at that time, rather harsher conditions appeared, what is indicated by very low abundance of biological indicators. How it is, after that, we tried to answer the question how our records fit to the general pattern in the Central America, general pattern of the conditions that were reported for the Little Ice Age in this region. And it's turned out that the general pattern in the region is not coherent. So in the north part of the region we analyzed, we have like the drier conditions. In the Yucatan Peninsula, in the north part, the records display drier conditions than during Little Ice Age. Here in this part, we have information that there were rather wetter conditions. So in the Belize, in the records from Belize, the records, sediment records displayed the dry beginning of the Little Ice Age and wetter conditions after that. So the data are inconsistent. Even the lakes that are close to each other, like Lake Petenica and Lake Salpeten, they display totally different signals at that time. Of course, it can be correlated with the geographical and climatic diversity of this region, because the lakes are very different. So we have in this region like the karstic lakes, like the tectonic lakes, volcanic lakes, and all of these lakes, they have different size. They have, of course, different depth, so they can be their sensitivity to the different environmental and climatic factors can be totally different. But actually, it turned out in the last paper presented by published by Obrist Farner, it turned out that even the records from the same cave, because from the Speleoten records, they display two opposite scenarios. So up to now, nowadays, the question about the reason of this incoherency in the records of the climatic changes during the Little Ice Age in Central America is answered, and we are not able to explain it. And after that, I would like to also mention that the time that were linked with the Little Ice Age in the Central America, it also was time of the decreasing of the population in El Salvador, what was related with the arriving of the conquistador from Spain. So epidemics caused that even 90% of the population died. So the next zone, so the next layer of the sediments, correspond with the beginning of the 20th century and recovery of the indigenous population. And here we can observe that gradual increasing of the trophy state, along with the higher content, higher share of higher abundance of the biotic of biota. So it could be caused by the higher input of the nutrients from the catchment, and yeah. And the most interesting, especially for me, was the uppermost layer from our core. So as you can see, we have totally different composition, especially in the diatom assemblage and cladocera assemblage. So suddenly, all species that the species that were dominated through the most of the core disappeared, and new taxa appeared, like the big, large-bodied cladocera, like Daphnia and Moina, and also new species significantly increased share of the species in the diatom assemblages. That is indicator of the turbulence water. So we were thinking, what could be the reason of such changes? In case of the cladocera, changes, so replacement of Bosminidae by Daphninidae could indicate likely disappearance of the fish, because usually the large-bodied cladocera are food for the fish. So when the fish disappeared, they could colonize the lake. And also Moina, so another large-bodied species that appear at that time, indicate also turbulence water. So what we figure out, actually we started to looking for the information, the local reports, and we found the information that after earthquake in 2001 in our lake, they observed change. Firstly, fish massive kill. So we have answer for the question about changes within the cladocera assemblages, and also change of the lake water color. So from the transparency into more green. So besides of the building clothes and so on, it seems that this earthquake had also significant effect on our lake. And this earthquake was very close to our study site, because it was located like the 30 kilometers far from our lake. And okay, you can ask me why this one was important. Because as you likely know that the El Salvador and general Central America is very seismically active region, and actually 1 month before this earthquake in February, they also recorded another earthquake that was caused by subduction of the coconut plate into Caribbean plate, but the epicenter of this earthquake was deeper, and the reason was different than the reason of the origin of the earthquake from the February. The earthquake from the February was caused by rupture of the fault zone, so its epicenter was very shallow, so have more destroyed effect on the ground. And for our curiosity, we also studied the surface sediments to see how our lake recovered from these dramatic events. And as you can see, again, the species that appear at the time just after earthquake appeared, and such taxa like Bosminidae back to the lake. Of course, also some new taxa appeared in the diatoms assemblages and also in the cladocera assemblages, which may suggest just partly recovery from the event. And thank you for your attention.
Okay, thank you, Martha, for the presentations. It's very interesting. Is there any questions from you? Okay, I just check the chat room. Or if you have a question that would like to deliver directly to Martha, please raise your hand, or you might be able to write down in the chat room. You also able to deliver your questions in Bahasa Indonesia, and then I will help to translate it to Martha. Okay. Any questions? Yes, in the chat. Oh, somebody need the link of presentations, and then probably I will put it later on. Okay. And then please, because I know that there is some lecturer from Syah Kuala, and then Palangkaraya, as well as Unsud, and then as well as the one that also study on paleolimnology from geology, Bu Anis and Pak Sasmita. Probably you have questions. Okay, while waiting their questions, Martha, how do because sometimes changes because of human activities or natural catastrophe happen probably in the same times, or probably there is a shift between human activities and then catastrophe. How is that reflected in the lake sediments using the Cladocera or other organisms, or probably what I mean is how to interpret which one is higher, human pressure, human activities, or catastrophe or natural?
This is a very good question.
While waiting, the audience asks your questions.
Actually, I'm. Okay, so this is a very good question. I'm not sure if I'm able to answer. Of course, we need to, especially nowadays, when the human pressure and effect of the dramatic natural events overlap very often. So sometimes we are not able to distinguish which one it's actually responsible for given changes in the environment. I think it's very good research question to study in the future. But yeah, what I imagine now, actually, usually, let's say that we need to take into account everything. So we need to integrate the data, not only from one proxy, and integrate also the local information, as I mentioned. And then we need to have overview of the situation of the given lake, and then we can try to figure out which factor was the most important in this specific case. But in general, what I can imagine in case of the anthropogenic factors, it will be rather long-term changes, yes. And in case like the earthquakes, it's a sudden event. So as you saw on the diagram, it was like they just reported in one layer in our core.
Okay, sorry, unmute. Okay, there is one raised hand from Pak Ardianor, please, from Palangkaraya.
Bismillahirrahmanirrahim. Thank you.
You still mute.
Assalamualaikum. Oh,
No, I hear you.
Okay.
okay.
Yep.
Thank you, Professor Tri. Dr. Mata, this is very interesting and lecture, yes, relating to your study in the Central America. So you use the carbon dating to estimate or to determine the past time of the age of the sediment. So the method is you use isotope, like. So I think this slightly expensive. So in Indonesia, we should go to Indonesian National Nuclear Energy Agency. We call Batan. Yeah. So maybe also Professor Tri have experience about this method. Yeah. So how do you estimate? What is the method you use? How you estimate the dating isotope?
Actually, yeah. Yes, it is expensive. I am not specialist in the dating at all. So what we did, of course, you need money for C14 dating, and this is the reason why we have only one C14 dating in our course, because this course was analyzed outside of any project. So everyone just was involved and we wanted to do something. So everyone did something. And this one was analyzed in the commercial laboratory, in beta laboratory. It's quite expensive. We did it on the extract of the pollens. But I know that, for example, in Poland, there are some less expensive laboratories. So if you want, I can send you links to these laboratories.
That makes sense. Thank you very much.
Yeah, the ones that usually can be developed, Pak Ardianor, is try to make a collaborations with the overseas universities. Yeah. Why Marta in Indonesia, in Semarang, because we have research collaborations, and then Marta will do some analysis that is unable to develop in Indonesia, because for dating at the moment in Batan, for lead to 10 for PP, I think it's still, I don't know exactly, but last times it's still broken and need to be fixed, and that is why it is difficult and need long time whenever we try to analyze in Batan, but as well we able to have collaborations, and then that is one of the solutions whenever we have kinds of these problems, Pak Ardianor.
Thank you.
Yeah, of course, yes. Collaboration is crucial, I have to say. There is many people that want to do something, so want to do science. So you need to just find people, talk with them, and yeah.
Okay, probably next time Pak Ardianor will invite us to work in Oxbow Lake, because in Kalimantan there are many Oxbow lakes, and I interesting to do that. 4 years ago, we have a core from Lutan Lake, and then I haven't had the result, but again, it's related to other results from another laboratories that until now we haven't, especially for dating. Marta, there is questions in the chat room from Agatha. Yes, professor also in limnology from Universitas Jenderal Sudirman.
So yes, thank you for this question. It's very good questions. Recently, I had just had one of the presentation about the multi-proxy, and I mentioned there that the thing we should avoid is to think that some biological indicators are more relevant than the other ones. But of course, I need to be honest. And Cladocera are not, let's say, the difficulties with the Cladocera is that there are not so many indicator species. So we need to try to assess the changes in the environment based on the changes within the whole community and based on the changes within the ratio of the planktonic and littoral taxa. So this is this advantage of the Cladocera, because, for example, as in the case of the diatoms, we cannot reconstruct exact the qualitative changes of the, for example, pH. And many research display that that Cladocera are very broad tolerance for the environmental changes. So it's quite tricky to reconstruct it, but we cannot obtain like qualitative data. And the advantage is for sure that they are very common in the lake sediments. Usually they are very preserved, and it's very easy to find them, and it's quite quick method. So you don't need to spend so much time with analyzing them, as in the case, for example, of diatoms. You have a limited number of species that you can find in the lake sediments, what is in contradiction to the diatoms, for example. So yes. And have you found a standard? In generally, in the literature, you can find information that one of the recent publications is saying that to talk about the environmental changes based on the Cladocera records, you need to count like the at least 70 or up to 100 individuals, not fossils, individuals. And for taxonomic purposes, you usually need like the 200 of individuals. I hope that I answered the question. If not, please let me know.
Bu Agatha, is there any other questions?
Oh, thank you very much for the explanation.
Yeah, it was okay for you?
Yeah.
Good.
Thank you.
Welcome. And you still have another questions from Anis? Yes.
So I will read the question maybe. The question number 1, can we use the same approaches that we apply on lake sediments to the older lake sedimentary rocks? Do you mean like the paleo lakes? If so, yes. In case of the coal, in general, for example, you can use this also for the peatland, so I guess for the coals as well. But I never tried it, to be honest. But the same procedure is doing for the paleo lakes. I'm not sure if you are satisfied with this answer. Okay, so I will go to the next question. So is there any specific treatment for the collected sediments to preserve the biological and geochemical properties of the sediments before we analyze it in the lab during transportation from the site to the lab? Yeah, usually during the transportation from the site to the lab, there is no special requirements, but immediately when you reach the let's say university or your research institution, it should be keep in the cooling conditions. Because because in the higher temperature, for example, some remains may be decomposed or and so on. So this is the mainly requirements in case of the lake sediments. And of course, if you collected the core, it's better to keep it like in this position, not on the horizontal one, to avoid the mixing of the sediments.
Bu Anis, is that already answer your questions, Bu Anis? Okay. So you have another question from Pak Taufik. Yes.
Very good question. Honestly, I don't know answer, but the thing, the thought I already had now is that maybe we should try to look at the type of the sediments we have that is correlated with the high content of the sulfur. Because from the volcanic activity, we will have very specific mineral composition and very specific sediments deposited in the lake. So this is what I would do during the analyzing of the volcanic and anthropogenic influence in terms of the sulfur content in the lake.
Pak Taufik, is that already answer your questions? Probably you have another opinions.
Yeah, I'm open.
Okay.
I have new thought. Maybe we could also do like the analysis of the stable isotopes of the sulfur in the sediments. I am not specialist in it, but I think so it could bring the answer because the different sources of the sulfur will have different isotopic signature. So maybe this is also the way to distinguish these two sources.
Yes. Okay. Yes. I think that for using the heavy metals like sulfur and then ferromanganese from the sediment, it's also able to help to reconstruct past conditions related to the volcanic or natural disaster, because I also have a data from some telaga at Dieng, and then it is there is a trend of increasing such heavy metals related to the timing when the volcanic activity is high. You are right, Malta. I have also the experience like that one. Okay, thank you, Pak Taufik. Probably any other questions for the audience or students? Okay. Hello. Any other questions? Martha, probably do you have experience relating the past conditions of the lake with the riparian vegetation. Do you have experience about that?
Like reconstruction of the riparian.
Yes.
I don't have experience. Like actually, based on the Cladocera assemblages, we can say in which period the submerged vegetation start to colonize the lake. So this is what we can do. If you would like to reconstruct like the exact species composition of the plants in the lake, you need to make macrophytes, macrofossils analysis.
Okay. Okay. I have to say that the Cladocera research is interesting, especially in Indonesia, because probably it is timing for us. Somebody probably that joined in this meeting, starting to work with Cladocera, because I don't know exactly who is the expert at the moment. So please let try to develop it, because Smarta have a lot of experience working on tropical Cladocera, and then it is starting a good starting to develop it. And then I hope somebody that interesting to do that. Probably we might be able to have collaborations, because honestly that to develop a good research, we cannot do everything by ourselves. We have to integrate it many data from many aspects, and then put it together, and then try to interpret based on a lot of data.
Yeah, it's true. It's true that the collaboration is one of the most important part of the research, especially in paleolimnology nowadays. But as Professor already mentioned, that there is time to develop subfossil Cladocera analysis in Indonesia. It is time for you, because nowadays there is no people who are working with the subfossil Cladocera in this region. So I really encourage you to interest in this topic.
Okay. I still open. We still have times. Is there any other questions from the audience? Okay. So before Marta give the last statement, I will try to resume that paleolimnology is combinations of paleontology and then ecology, as well as limnology, and it is working with chemical and then physical as well as biological aspect that preserve in the sediment. And then based on what happened and record from the sediment, we able to reconstruct past conditions that whenever we already know about what happened in the past, is that naturally, or it is more human activities that give high pressures in the environment, we might be able to develop suitable management related to our lakes. That it is important, because we have many lakes in Indonesia, and some of the lake is still pristine, but many lakes have problems due to eutrophication, for example, or sedimentations related to the high slopes of the catchment area. But whenever we are talking about lakes, it doesn't only talking about water body in the lakes, but also what happened in the catchment area. So that is the important things, and why we able to reconstruct what happened in the catchment area, because it will be come down to the lake and then recording in the lake sediment. So that is that I can resume from Marta talks, and then there is the last things is there is a challenge for us to develop another approach, not only using diatoms, not only using pollen, for example, but also able to use Cladocera. And I had involved yesterday that working with Cladocera is challenges and interesting because Cladocera is bigger than diatoms, and then it is that is why it is able to distinguish for example from planktonic or littoral, and it also will able to talk about past conditions. Okay, Marta, probably before I deliver to Pak Panji, is that any last statement that you want to share?
Yeah, so not only Cladocera, but also, for example, like the chironomids, there is still no people who are working with this in this region. And I would like to also add that the paleolimnology is super important, especially in nowadays when we observed, as Professor already mentioned, the deterioration of environmental conditions in the lakes. And the paleolimnology can provide because good management of the lakes, so good plan for the restoration needs knowledge about the lakes, like natural conditions, let's say, so needs to have like the baseline. So the paleolimnology is the one of the type of the research that can provide us an information how the lakes looked like before the human started affected on this lake. So they are also very important for the restoration purposes and management of the lakes. And from my side, I think so. It's everything. I would like to thank you very much for today. And if you have any question, feel free to write me anytime. I will write my email on the chat. So feel free to contact me. Thank you very much.
Thank you, Marta, again, and I hope our collaboration will strengthen. And then thank you for all of you that attend these lectures, and then hopefully it motivate you to develop Cladocera or chironomid to the paleolimnological aspect. And thank you for the Dean and International Office from Faculty of Science and Mathematics Undip that held these lectures. And also thank you for all of you. I hope all of you have a succeed and have a nice day. Thank you. I deliver to Pak Panji. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Tri Retnaningsih, Professor Tri Retnaningsih. And thank you very much also Dr. Marta for the very enriched discussion and also enriched lecture for today's visiting lecture sessions, and also Professor Tri Retnaningsih for leading the discussion sessions and also moderating the whole session. And to close this morning sessions, we would like to give our sincere appreciation to Dr. Marta. So we already provide a certificate as a visiting lecturer, and hopefully we will give it to you directly. And perhaps the certificates already ready or not, Mr. Oki? Perhaps you can show it to us. Or otherwise, we will give it to you in the room also. Okay, and without further ado, thank you for all the participants who already joined this morning sessions, and also thank you Dr. Marta and also to Professor Tri Retnaningsih who giving the discussion as also the lecture for this morning. We hope that this visiting lecture sessions will give us a broader idea and also broader knowledge to do research and also collaborations in the future. And we hope to see all of you in the upcoming visiting lecture provided by the Faculty of Science and Mathematics. So we will close these sessions and thank you for joining us. Terima kasih. Good morning. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you. Terima kasih.
Bye-bye.
Okay.